Hi, it's Dr. Franco Cavaleri coming to you today for another session of Potential Within. Today we're going to talk about a molecule that is completely overlooked, but can change your life profoundly in terms of all these disorders and dysregulations that we may be experiencing, that we may be attributing to age. And not only age, but we start to see this movement of disease, propagating disease, because of environmental factors, dietary factors, the processing of our diet, all these things we're seeing and hearing about in the North American lifestyle that's contributing to disease that we cannot evade. But we can make some serious changes by going back to basics and actually focusing on this small molecule. Life can change for you with the application of what I'm about to talk about. It relates to the microbiome. We've talked about the microbiome in previous episodes, but I'm going to hone in and be very specific about a postbiotic. Now, you've heard of prebiotics, the fiber that the microbiome will use to make byproducts that are important, metabolites, important for your metabolism and various aspects of insulin function, cognition, inflammatory controls. I'm going to talk about why and how this small molecule, this postbiotic, not a prebiotic and not a probiotic, but a postbiotic, a common term that's becoming, that's trending in the last 5-6 years. The one in particular I'm going to hone in on is butyrate. Now, let me tell you about butyrate. This is a short-chain fatty acid. Butyrate is made up of 4 carbons. There are other short-chain fatty acids, propionate and acetate. And the propionate is a 3-carbon chain fatty acid, acetate 2-carbon chain. What's important about these postbiotics that we've come to learn today are life-changing? And I'm going to kind of scale this back a little bit and talk more about the fact that the microbiome, our diets, our lifestyles, have compromised the microbiome throughout the body, in particular the colon, the large intestine, and throughout the intestines. And I want to highlight that the concept of microbiome is not isolated just to the colon, large intestine, or the intestinal tract in general, but we have specific microbiomes that grow in orifices of the body, on the surface of the body, and they coexist to support health. Now, the one thing that we got to keep in mind is that this microbiome, or the variety of microbiomes that live symbiotically with us, we depend on them. We evolved depending on them. And when I talk about the probiotics, the postbiotics, the short-chain fatty acids that help relieve us of many disorders that we may be plagued with, it's not so much that they are therapeutic. It's more that we depend on their existence in order to function normally. So we co-evolved with these cultures, these microbial cultures, and became dependent on the byproducts that they produce. And the dependence goes as deep at as the cellular level. I'm going to talk a little bit about butyrate and the gut-mind connection. And we've heard about this, the gut-mind connection. And it comes at different levels and different types of connectivity, from serotonin to the probiotics and the postbiotic secretions that these probiotic cultures generate. Butyrate. I've studied butyrate for about 35 years. In fact, I've used it when I trained for bodybuilding contests long ago. And it's a disgusting compound that tastes terrible, smells terrible, But I recognize the value based on the science. At the time, there was very little known about it 30 years ago. In the last 15 years, we have studied it in the laboratory extensively to determine the pharmacology and how it affects various aspects of metabolism. And the findings are profound. Profound. So butyrate, this molecule, is produced generally by the probiotic culture in the colon, and it does creates an environment in the colon, the pH specifically, that voids pathogens. Pathogens don't like to live in the environment that's butyrate loaded. But that environment also serves as an amazing cultured and nurtured environment for the probiotic culture. So by supplying butyrate in one form or another, what we do is we generate the environment that propagates an exponential health for the probiotic culture and voids pathogens. And so the key thing to think about is that many of us have compromised microbiomes to begin with. So this butyrate secretion is insignificant or compromised. And then, as we know, we have this epidemic of metabolic syndrome. And what does that mean, this metabolic syndrome? Metabolic syndrome is really a compilation of many disorders that center on insulin resistance, that involves a domino effect, a cascade that creates metabolic inefficiencies, including escalated inflammatory activity, or what we might call subclinical inflammation you don't really feel, but nips away at your body and increases the risk for disease, primarily, as we know, and would be logically assumed, type 2 diabetes, type 3 diabetes, obesity, which is related to that metabolic dysfunction. And so when you begin to study butyrate in the context of metabolic syndrome, you start to see why it's so valuable for the body, why the body depends on it. And when it's not available, because the fiber that you do need in your diet is not available for the probiotic culture to consume or ferment, more specifically, to create these byproducts, or the microbiome itself is compromised because of glyphosates and other pesticides and things in the environment that are toxins, including the high glycemic index diet, which can cause toxicity throughout the body in terms of pH anomalies. And so when we restore the microbiome, and this is very difficult to do, it's actually, it's not as easy as just taking a probiotic supplement. When you restore it, you begin to see resolution of multiple conditions that are related to butyrate insufficiency or deficiency. And so one thing to keep in mind in relation to what I'm talking about as I comb through the specific cellular components and metabolic pathways that butyrate really integrates into, is to really think about, if butyrate wasn't doing at a cellular level each time I speak to its activity, how dysfunctional your cells could be. And that dysfunction then results in disease. And we've known for some time that many disorders, cognitive, cardiovascular, the cardiomuscle, skeletal muscle dysfunctions, are highly and intimately related to mitochondrial dysfunction. Mitochondria is the powerhouse of the cell. And that's where your ATP is generated for energy. And you can imagine, if the mitochondria aren't working well, a lot of things start going wrong. Forget maximal performance as an athlete, because the mitochondria is central to creating the energy for load, force, speed, fast twitch, and slow twitch. Cardiovascular health, every single cell depends on ATP. And so you can see, if the mitochondria is dysfunctional because butyrate is not available in sufficient quantities, your entire body becomes to hiccup. It just doesn't function the way it's supposed to. And so the supplementation with butyrate then become a therapeutic compound to treat all these conditions? Or are we simply just supplying butyrate in supplemental form because we first damaged the microbiome and made butyrate insufficient in terms of its availability prolifically and peripherally throughout the body. So let me give you some context in terms of how butyrate affects the cell and how it relates to insulin efficiency. Butyrate actually activates a kinase called AMPK. And for those who are in the know in terms of cellular activity and in relation to cellular performance, AMPK is like a sensor, or it's a button, where once activated, it facilitates insulin function. And if it doesn't become activated, glucose transport, insulin function, cellular energy, and even the burning of fat as a primary energy source becomes compromised. And so here's one feature alone that profoundly affects the energy management and including macronutrient usage for ATP synthesis for the production of energy by the cell, AMPK. If it's not activated, not phosphorylated to be fully active, then the cell is compromised in terms of its response to the nutrients you consume. Right there, big factor in the contribution to insulin desensitization, in other words, type 2 diabetes, because the desensitization of insulin as it progresses then becomes type 2 diabetes once you've lost sensitivity of insulin at a level that's type 2 diabetic. However, we all, as we age, encounter pre-diabetic stages of insulin resistance that create various degrees of metabolic dysfunction. It's one of the reasons why it's harder to keep fat weight off as you age, and build muscle as you age, because insulin is required as a signaling mechanism to cause nutrients to enter the muscle cells, skeletal muscle cells, in order for them to create protein or protein synthesis to be activated. And so if insulin desensitization or insensitivity begins to occur as you age, then the body needs to produce more insulin in response to each meal to compensate for the desensitization. And more insulin means that energy substrates like fat, excuse me, and other macronutrients, they, because of this insulin overload, end up being pushed to storage sites, and it's hard to pull that fat from the adipose tissue to be utilized as an energy substrate. You can see, going into a workout, for instance, when you're trying to burn fat and you have insulin resistance, it's harder to use fat as a primary energy substrate during that activity, compared to if you can improve insulin function and therefore then enhance metabolic activity that uses fat as a primary energy substrate during activity. Butyrate improves and supports insulin function. And it's not that it's miracle. It's because our body is designed to depend on butyrate for insulin sensitivity. And so when it's absent, the body hiccups. Same with cognitive function. I published a paper a while back on butyrates, including beta-hydroxybutyrate, which is a ketone, and how it functions in the central nervous system to support cognition. And, butyrate itself can enter the mitochondria and become butyryl-CoA to run through the electron transport chain and itself be a substrate for ATP. And here's the interesting thing. When this fat, just like other fats we've spoken of before as ketones, when they enter this cycle in the mitochondria to generate ATP from that substrate, the mitochondria works more efficiently than it can with glucose, with a reduced consequence of oxidation. The other thing that's noticed in the cell when butyrate's readily available is that various endogenous antioxidants are enhanced, like superoxide dismutase, which means if you're active especially, or you're fighting disease, you are more prone to this oxidation and you need a response by this endogenous antioxidant load. Butyrate supports that. Butyrate itself is a fuel, as I said, especially for the colonocytes and the colon, and it helps to create very healthy barrier against microbes that may travel systemically from the colon. A lot of amazing things that butyrate does. Butyrate acts as a histone deacetylase inhibitor. What the heck is that? Well, epigenetic activity and influences on our genes tend to cause those genes to become less efficient than they were when we were younger. And one of the primary places that butyrate has an effect on the genome is on genes that regulate inflammation. And as these acetylation points aggregate, they tend to interfere with the balance and controls at a genetic level of inflammatory control. In particular, the regulation of the NF-kappa B signaling pathway. Now, during my PhD thesis work, the NF-kappa B signaling pathway was central, and I studied many anti-inflammatory nutraceutical isolations in the context of inflammatory activity and anti-inflammatory controls. And butyrate became a central interest for me because for any molecule I studied in terms of NF-kappa B signaling inhibition, and that's anti-inflammatory activity, butyrate enhanced it. And it's an interesting concept because when you see a regular, frequent, and reliable change in anti-inflammatory activity for any drug, whether it's natural or a pharmaceutical unnatural chemical, and you see a reliable increase in activity for anti-inflammatory drug targets, you have to wonder why. Well, if you're working in an in vitro environment, butyrate's not available because we're not supplying it, because butyrate's only available in the context of a systemic body where the microbes in the probiotic culture are producing butyrate. And in that context, you can only assess butyrate's value, whether it's increasing the anti-inflammatory activity, when you add it to the isolated tissue and or cell culture. So then, at that point, we begin to realize that there's a critical synergistic application, but it's not synergy as a drug. It is actually a biological dependence on butyrate's activity at a subcellular level. We begin to see how, in our research, butyrate supports the efficiency of the mitochondria in terms of the electron transport chain. And many of you have probably studied the electron transport chain in high school, where that energy transfer results in the production of ATP. And so we see butyrate improving the mitochondrial activity to the point where you see mitophagy occurring where mitochondria are damaged. What's mitophagy? Well, it's analogous to autophagy. And autophagy is when the body, or the cells in particular, become very effective at clearing debris, dead organelles, damaged organelles, and things that don't belong in the cell that interfere with biological activity. Well, in the context of mitochondrial dysfunction, butyrate facilitates at a genomic level the clearance and the removal of mitochondria that are inefficient and the induction of biogenesis of new mitochondria. Now, that's amazing. So the interesting thing here is that butyrate is an essential factor that runs around creating efficiencies in your cell. And when not available, you develop disease. So the question is supplemental butyrate then a great therapy? Or is the lack of butyrate the cause of disease because it's essential for healthy function? And it's the latter. However, I'm going to continue on our analogies and reporting as we study different compounds associated with metabolic efficiency, including thermogenesis, the activation of uncoupling protein and the brown fat. We've done a lot of work on it. I talk a lot about it in Potential Within, the book I did in 2000. In year 2000, this was published. It's still relevant, although we've advanced our research to be more pinpointed in terms of the mechanisms that we found that were reported here. And this was kind of a prologue to my PhD work. now we've kind of refined the mechanisms to be more specific and more pointed in how we manipulate and utilize natural compounds to be pointed at the drug targets. And I say drug targets, I mean targets within our cells that are responsive to the compounds that then relate to the treatment of disease indications. And so our curcuminoids that we've been highly effective at engineering to be more effective than regular curcumin, we recognize there too, complexing butyrate to the curcuminoid improves the anti-inflammatory effect of the curcumin. And there's two reasons for that. And I give you the specific example because it kind of gives you an idea of how this works globally throughout the body. By complexing the butyrate to the curcuminoid, what we do is we also offer an opportunity for the probiotic culture in the colon to become healthier. And the one thing that's critically important is that culture helps metabolize a lot of the nutraceutical compounds, especially herbal extracts. The isolated actives within those extracts, they are metabolized by the probiotic culture. And the metabolites that are created are often the actives that go into the body systemically to affect you in the way that you believe is positive in the context of the herb, whether it's green tea, whether it's curcumin, ashwagandha. And so the natural thing would be then to complex those to butyrate in order to facilitate their activity and heighten the the activity you expect to get from those herbs. And we've done that time after time. And we've shown how butyrate not only acts as a facilitative carrier, but it supports the microbiome so it's healthy enough to metabolize the compound that you're taking and then create the metabolite that the body can use effectively. Now, this discussion is, really, it's designed by me to try and, with no script, but to try and really highlight the value of this short-chain fatty acid. There's a lot of research independent of that which we've done that shows if butyrate levels are actually low in the brain, it contributes to mood disorders and behavior dysfunctions similar to autism and what used to be called Asperger's, which is kind of borderline autism, depression, anxiety. Why? Butyrate's not a recognized treatment protocol for that. But again, the lack of butyrate then causes central nervous system anomalies that result in what we call disease that then we treat with pharmaceutical drug rather than treat the root of the cause. And the root of the cause generally is the microbiome. And, a lot of us have really gotten into probiotic treatment with probiotics to kind of restore the microbiome. It's not that simple. It's not as simple as taking a, 20 million probiotic culture capsule, even a 1 billion or 2 billion or 3 billion. We had a great discussion with a group of doctors last night. And when I brought this up, they were all concurring because they too were using the very high doses of probiotics to treat various conditions that we're talking about today. And why? And how much did they take? And here's how much they were using, how much I use, between 100 billion to 300 billion a day in order to correct the compromised microbiome. And I would go in and take the 100 billion, and one of the medical professionals was talking about using 300 billion in some of his patients for 3 to 5 days, for 3 to 5 weeks. What I normally do is I'll take about 100 to 200 billion 5 or 6 days in a row. And then I cut that to about 50 billion for the rest of the month. At the end of the month, I start over again, 200 billion a day for 5 or 6 days, and then go down to 50 billion. And the idea is that you have to keep inoculating the colon, because if you don't, the body wants to go back to what it's used to. You see, we all have, based on lifestyle, based on what we were inoculated with when we were born. If you were born and you came out of the vaginal canal, you probably have a better microbiome than those who did not, those from cesarean. So there's a lot of things that contribute to the development of that microbiome, including your food, your environment. I mean, if you go to someone in a different country, they're always going to have a different microbiome proportion. And it works for them because their body evolved with that microbiome to function for them. And if they move away to another country over time, that microbiome changes. And so if we want to re-inoculate the microbiome, we have to run heavy doses and use them for a long time in order to keep the body from reverting back to what it was used to. And then reverting back to what it's used to may be the very thing that's contributing to your diseased state. For one reason being that butyrate content is not being produced the way it should. And therefore, this is our rationale then in drug development, and we've got several patents in the context of this, is taking that butyrate molecule and accompanying it by the targeted drug treatment, and it's enhanced every time, simply because our cells have evolved genomically to depend on butyrate because we don't manufacture it. It is manufactured by the probiotic culture that has symbiotically embedded itself within our beings. We have more probiotic cells in and on and around our body than we do mammalian cells. There's a reason for that. These are not dysfunctional cells. These are cells that support us symbiotically and we support them. And so there's a reason for it. And the reason is, again, our health depends on it. Butyrate supports AMPK, and through that supports insulin function. If butyrate's not available, insulin function declines faster with age than it would for someone who's healthy. Inflammatory control depends on butyrate availability and the effects of butyrate on your genome. That's how deep the effects are. The effects on how your genes are accessed for transcription of various proteins that are involved in controlling cell function. That butyrate's involved in the gut-brain connection. It actually literally migrates into the brain and affects your neurons. Butyrate itself affects the evolution of macrophages and T-reg cells of the immune system. And those T-reg cells are critically responsible in managing inflammation, whether it goes up and down in the cases of autoimmunity. And in fact, the failure of butyrate availability or production and the compromised butyrate availability is a factor that contributes to the failure of the immune system to control itself in the context of auto-inflammatory conditions or autoimmunity, which are two separate classes of conditions, autoimmunity and autoinflammatory conditions. And so you think about the prevalence of these diseases related to immune dysregulation, the prevalence of diseases relating to insulin dysfunction, and those related to mitochondrial dysfunction. And all of these targets are associated and dependent on butyrate availability. And therefore, that's why we've gone down the path of complexing our treatments to butyrate to make sure that optimal efficiency of the metabolism is put in place so that any treatment that you're trying to apply has a chance to penetrate and deliver an effective result. I'm going to cut the butyrate discussion here, but I think this is something that we're going to talk about in different contexts over the next few episodes because it is such an important subject matter that I think everybody needs to get this profoundly and relate it to the microbiome, which is becoming more and more known, studied, and recognized as central to metabolic health. Thanks very much for the short episode. See you in the next one.